So let us start with today's episode of biological classification. So what are the learning objectives of today's lesson? So at the end of this lesson, we will be knowing about kingdom Monera, its characteristics, what are the animals or organisms which are falling around, under this group. We will be knowing about kingdom Protista. Kingdom fungi, it's a bit vast part, so I have taken it in the next video, and we are going to discuss about the virus, viroids and lichens. Okay, so that also in the next video. So in this video only we will be discussing about kingdom Monera and kingdom Protista. Now first let us discuss about the basic of classification. So since the dawn of civilization, there have been many attempts to classify these living organisms. So it was done very instinctively, not using the criteria that were scientific, but born out of need to use the organisms for our own use for food, shelter and clothing. Now first came Aristotle. So what did he do? He divided these organisms under the basis of simple morphological characters. He divided the plants into trees, shrubs and herbs. Then what he did? He divided the animals into two groups, that is one with the red blood cells, the anima, and the other one with the without the red blood cells, that is anima. So Aristotle was the first one to divide the characters plants into trees, shrubs and herbs, and animals into two groups, one with red blood cells, that is anima, and one which did not have red blood cells, that is anima. So this was the classification done by Aristotle. Then Carl Linnaeus, he came. What he did? He divided the classification into two kingdom classification. He divided simply organisms into plants and animals. So Plantae and Animalia were the two groups he told. But what happened? The system is actually, he did not distinguish between what? Eukaryotes and prokaryotes. He didn't mention the difference between unicellular and multicellular organisms, and he didn't distinguish between photosynthetic animals, that is the green, blue-green algae and all, and the non-photosynthetic animals like the fungi. So his classification was also having so many flaws. That is, he did not distinguish between these characters like eukaryotes, prokaryotes, unicellular, multicellular and photosynthetic as well as non-photosynthetic. So he had also and many flaws in his classification system. Now gradually classification system for the living system underwent several changes over the time. But the plant and the animal kingdoms have been constantly under different systems. So always we have divided plants and animals under different systems only. So how we came to this five kingdom classification? This is now the question. So who discovered this five kingdom classification? Now this five kingdom classification was discovered by R.H. Whitaker. In 1969 he proposed the five kingdom classification system. So how he proposed? He mainly classified them using certain important things. First is the complexity of the cell structure, its body organization. So first was the cell structure, second was the body organization, third was the mode of nutrition, that how they are dependent on what food they are eating, like autotrophs or heterotrophs. Either they are making their own foods or they are dependent on the others. So mode of nutrition was another factor. Reproduction was another factor, that is asexually reproducing or sexually reproducing. And the last part was phylogenetic relationship. So he divided the animals into animals or plants into five kingdom classification, like depending on these five structures, like the complexity of the cell structure, the body organization, then comes the mode of nutrition, then comes reproduction, and then comes the phylogenetic relationship. So what is this phylogenetic relationship? We should know this, right? This is the study of the evolutionary development and the history of group organisms. So this is the phylogenetic relationship. Now we will see the characteristics of the five kingdom classification. So what are the main characteristics of five kingdom classification? Now here we should know on what basis R.H. Whitaker divided this five kingdoms. So he told that characters, what we have already told, that the cell type, The cell wall, the nuclear membrane, the body organization and the mode of nutrition are the 5 main things or the characters on which he divided the 5 kingdom system. So when we are taking cell type, what comes here? In Monera, the cell type is prokaryotic cells. Okay, so it is prokaryotic cell, whereas in Protista, in fungi, plantae and animalia, they all are eukaryotic cells. Okay, in Monera, it's the prokaryotic cell, whereas Protista, fungi, plantae and animalia, they all are eukaryotic cells. Now next comes the cell wall. Now the cell wall is non-cellular in Monera, whereas in Protista, in some that is present. In fungi, it is present, but it's without cellulose. In plantae group, there are cell walls with cellulose. That is, cell wall is very prominent in plants. But in animalia, there is no cell wall. That is, absence of cell wall in animalia. Now next comes the nuclear membrane. So nuclear membrane, it is absent in Monera, but it is present in Protista, fungi, plantae and animalia. So in nuclear membrane, it is totally absent in Monera group. It is present in Protista. It is present in fungi, plantae and animalia. Now when it comes to the next point, that is the body organization. So how we are defining this body organization? Now Monera, they all are cellular. Protista also is cellular. But in fungi, when it starts coming, it is multicellular and it is having loose tissue. And plantae, they are having both tissue and organ. Either tissue, we can say plant tissue or organ is there. Animalia, they are having tissue, organ and as well as organ system. So it's more complex than Monera, than Protista, than fungi, than plantae. Plantae is having tissue as well as organ, but in Animalia there are tissue, organ and organ system. Now what is the mode of nutrition? Now when we come to this mode of nutrition, according to that we can say that Monera, it is autotrophic, it is chemosynthetic and photosynthetic and heterotrophic as well. And we can say that it is parasitic also. Now when it comes to Protista, it is autotrophic, that is, it is photosynthesis and photosynthetic and heterotrophic. Next in fungi, it comes out heterotrophic, saprophytic and parasitic. Next to plantae, it comes autotrophic. Autotrophic, it is photosynthetic. When it is autotrophic, it is photosynthetic as well. You know, right? When it comes to Animalia, it is heterotrophic. That is, it is depending on holozoic and saprophytic nutrition. So is it clear? So when we are dividing it upon the mode of nutrition, Monera is different, Protista is different, fungi is different, plantae is different, whereas Animalia is different. So Animalia, we can say that Animalia is always dependent on others for their making of food. That is, they are having the heterotrophic mode of nutrition or saprophytic. Okay, autotrophic, they are photosynthetic. Heterotrophic, we can say that fungi, they are also dependent on others like saprophytic as well as parasitic. Now autotrophic, photosynthetic and heterotrophic, Protista, in that we can say it's like that. And in Monera, it is autotrophic, chemosynthetic as well as photosynthetic. So this chart, please learn it properly. So it will be very much easier for you to learn on which characters the 5 kingdom classification has been divided. So Monera, Protista, fungi, plantae and Animalia. So today we are going to start from kingdom Monera first. Now first, under this 5 kingdom classification, we have taken kingdom Monera. So first comes kingdom Monera. So what are the main organisms which are under this kingdom Monera? The bacteria is the sole members of this kingdom Monera. So there are abundant microorganisms. So they are the most abundant of microorganisms, that is bacteria. So they are found in huge numbers in nature. So bacteria, how we can say that they occur almost everywhere. Hundreds of bacteria are present in a handful of soil. So they can live in extreme habitats, as they can live in hot springs, they can live in deserts also. We can see that they are living in snow as well as deep oceans, whereas very few life forms can survive. So many of them in or on the other organisms, they are staying as parasites also. Now this bacteria, when we are dividing this bacteria, we are grouping them under 4 categories. So what are the 4 categories on which the bacteria is divided? First one is the spherical or the coccus shaped bacteria, that is the plural will be cocci. Next comes the rod shaped bacteria or the bacillus shaped, that is the bacilli, we can say that the plural is bacilli. Next comes the coma shaped or the vibrium, okay, or we can say the vibrio, which is the plural is the vibrio. And last but not the least is the spiral, which is the spirillum. Okay, so it is called spirilla also. Now, though we can say that the bacterial structure is very simple, the spherical, rod-shaped, comma shaped like vibrium, we can say that spiral or spirillum. Now, these their behavior is very complex. The structure, though it's very simple, their behavioral approaches are very, very complex. So, compared to many other organisms, we can say that this bacteria, they show most extensive metabolic diversity. So, we can say that bacteria, they are showing the most extensive metabolic diversity. Okay, is it clear? So they are showing the most extensive metabolic diversity. So what we can say that some bacteria, they are autotrophic, that is they can synthesize their own food, whereas others, they are heterotrophic, that is they depend on others for their own food. So they may be photosynthetic autotrophic, or they may be chemosynthetic autotrophic. Okay, so the vast majority of bacteria, actually they are mainly heterotrophs, and they depend on the other organisms or on dead organic matter for food. So they are either photosynthetic autotrophic or they are chemosynthetic autotrophic. But the majority, what we are saying, they are heterotrophs, that is they depend on the other organisms or on dead organic matter for their food. Is it clear? Now we are moving on to the different types of bacteria which are under this kingdom Monera. So till now it is clear to all, bacteria is the sole member of Monera group. Bacteria, they are classified under 4 different distinct structural features. There are abundant microorganisms in this bacterial group. They are either autotrophic or heterotrophic. Just a quick revision, and I have made the notes in very simple manner so that you will learn each and everything. And the heterotrophs, they depend on the other organisms or on the dead organic matter. Now we are going to discuss about the main types of bacteria which are under this kingdom Monera. Let us start. Now under this Monera category, the first type of bacteria which we are taking here is the archaebacteria. So archaebacteria is the first category of bacteria. So these bacteria, actually why they are special? They live in extremely harsh conditions, that is they can live in extremely salty areas. Those bacteria, they are living in salty areas are called halophiles. Those which are living in hot springs, they are called thermoacidophiles. Now some bacteria, they are present in marshy areas, they are called methanogens, and some are present in the gut of ruminant animals such as cows and buffaloes for the production of methane or biogas from the cow dung of these animals. Okay, so they are present in all type of extreme conditions, either too salty or too hot or too cold, like that only. They are present in the gut of ruminant animals also, like cows and buffaloes, for the production of biogas. Okay, now different cell wall structure is they are having a different type of cell wall structure, that is mostly rigid cell wall they are having, which helps them to survive in this type of extreme conditions. So because of their cell wall, they are compatible to stay in harsh conditions. So next to this category, we will move on to eubacteria. Now this eubacteria, it's also called true bacteria, eubacteria. The other name is also called true bacteria. So how they are characterized? They are characterized by the presence of rigid cell wall. So these eubacteria, they are having rigid cell wall. They are characterized by rigid cell wall. So, and if they are motile, they are having a flagellum. And this, they are also referred to as cyanobacteria. That is cyanobacteria. Which one is that? That is blue-green algae which is having chlorophyll and has a similar, which is acting similar to green plants. So cyanobacteria, that is blue-green algae which are having chlorophyll, they are acting similar to green plants. That is they are autotrophic. That is they are photosynthetic autotrophs. Okay, so what are they? They are unicellular, they are colonial, they are filamentous, and they are mainly present in fresh water or marine water, or they are called terrestrial algae. Now how they are found? They are found in colonies and which are surrounded by gelatinous sheet. So what does this gelatinous sheet mean? So that is a slimy kind of sheet they are covered with. They are found in colonies, in groups they are found. They are not found singly. So in a single, you can't find eubacteria, single eubacteria. They'll stay in colonies, they're like group, and they bloom in polluted water bodies. So if you are going through the Archaebacteria, eubacteria, these characters, you will learn from each and every topic how I am teaching. The notes I am also giving in this only. So if you are learning these notes, it will be very much easier for you to remember how they are representing themselves in this kingdom Monera. Now, what they are doing, they are fixing atmospheric nitrogen in specialized cells which are called heterocysts. Okay, so two main examples are nostoc and anabena. So here this example is of nostoc. So this nostoc, what they are doing, they are having heterocysts. So this dark part, these are all heterocysts, okay, which are helping to fix the atmospheric nitrogen. So these specialized cells, they are helping to, heterocysts are helping them to fix the atmospheric nitrogen. Now chemosynthetic autotrophs are also there. What they are doing, chemosynthetic autotrophs, they are oxidizing the inorganic substances such as nitrates, nitrites and ammonia, and they are using the released energy for their ATP production. So they are oxidizing the nitrates, nitrites and ammonia, and they are using the released energy for the ATP production. Now these eubacteria, they are playing another important role in recycling the nutrients like the nitrogen, phosphorus and iron and sulfur. So they are also doing this, they are also doing that. They are also recycling the nutrients like nitrogen, phosphorus, iron and sulfur. So this is how they are recycling the nutrients. Now next comes in this category eubacteria. Under this category only we are having the heterotrophic bacteria. So these are all photosynthetic autotroph or chemosynthetic autotroph. That is, they are able to make their food on their own. Now we move on to the category that is heterotrophic bacteria. So let us see. Now comes the second category. In this eubacteria only we are having heterotrophic bacteria. So heterotrophic bacteria, it is mostly abundant in nature. This type of bacteria, they are most abundant in nature. Now the majority of these type of bacteria, what they are doing, they are decomposers. So they are all decomposers and they are having significant importance on human affairs. So what they are doing, they are helping us in making curd from milk. They are helping in the production of antibiotics. They are helping us to fix the nitrogen in legume roots. Now there are several pathogenic bacteria also which can cause damage. So what is pathogenic bacteria? Which are causing damage to the human beings like crops, the farm animals and the pets. So they are having both useful, beneficial effect as well as they are having harmful effect also. They are acting as pathogens also. Now we know that several bacteria like cholera, typhoid, tetanus, citrus canker, they are all causing diseases, like they are disease causing bacteria. So what is their mode of reproduction? So we know that on this characters only, the cell wall structure, the type of nutrition, the mode of reproduction, the mode of nutrition, on this basis only R.H. Whitaker, he divided all the 5 kingdom classification. So for each and every category we should learn all the characters in each and every kingdom. So Monera, Protista, fungi, plantae, animalia, in all we should learn each and every characters. So they reproduce by means of fission and under favorable condition, but when in other cases when their conditions are not so favorable, they are producing spores. That is a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacteria to other. Now there are certain other organisms like mycoplasma, which are organisms that completely lack cell wall. They are very small and they are known to survive without oxygen. So mycoplasma, it is known to survive without oxygen. They lack the cell wall and they are pathogenic in animals as well as plants. So this is all about this eubacteria, or what we can say that the heterotrophic bacteria as well as the Monera group. Now we will move on to the next group of the Protista. Now starting with kingdom Protista, what are these kingdom Protista? They are mainly single celled eukaryotes and they are placed under this Protista group. Now here the boundaries are not well defined. Actually, this Protista group, we can say that this eukaryotes, but their boundary is not mainly well defined, and their members are mainly aquatic in nature. Is it clear? So they are aquatic, and they being eukaryotic, the Protistan cell, what they are doing, they are having well defined nucleus. They are having other membrane bound organelles also within the cell. Now some of the cells in this Protista group, they are having flagella or cilia. Reproduction, what is the type of reproduction? They are involving cell fusion and zygote formation, that is both sexually and asexually they are reproducing. So under this Protista group also there are several different types of animals. So they have been divided into 5 other subgroups. Now the first one is chrysophytes, then comes dinoflagellates, then comes euglenoids, then comes slime molds, then comes protozoans. So 1, 2, 3, 4, 5 categories under this kingdom Protista we have divided. So we will learn each and everything in details in your 11th standard now, because in previous classes also, in 9th also you have studied about this classification. So you have studied a bit about each and every group, their characteristics and all, but you have not studied about each and everything in detail. So this is in your 11th standard. For your NEET exam, for your 11th board exams, you need to learn each and everything in details. So go through it, and please don't forget to subscribe to my channel to keep on getting the updates. So on the kingdom Protista, the first one is the chrysophytes. So what are these chrysophytes? Under this category, we can take the diatoms and the golden algae or the desmids, which are found mainly in fresh water as well as in marine environments. So what can we see them with our naked eye? No, they are mostly microscopic, and they float passively in water currents and plankton, and most of them are photosynthetic. So they are passively floating in water currents. They are also called plankton, phytoplanktons or zooplanktons. We have studied them, and most of them are photosynthetic. Now what are these diatoms characteristics? They are having cell walls forming 2 thin overlapping shells. So what are they doing? What is the characteristic which is helping them to fit together in a soap box? So walls are embedded with the silica, and they are indestructible. So walls are embedded with silica and they are indestructible. They have left behind a large amount of cell wall deposits in their habitat, and as they have deposited, these cell wall deposits have been accumulating for many years. So this accumulation has caused the diatomous earth. So diatomaceous earth, sorry. So this accumulation of the cell wall deposits in their habitat is called diatomaceous earth. Okay, this term is very important. If the question comes, what is diatomaceous earth? So what is that? That is after leaving behind large amount of cell wall deposits for a large number of years. That thing is called diatomaceous earth. Now being gritty, this soil is actually used in polishing, filtration of oils and syrups. Now diatoms, these are all the chief producers of ocean. So these are planktons, these are chief producers of oceans, and as they are being gritty, this soil that is used in polishing, filtration and in making syrups and all. So let us move on to the next category, that is the dinoflagellates. Now here you can see the diagram of this also. So this is, you just refer to this diagram of this chrysophytes. This is the diagram. Now let us move on to the next one. So dinoflagellates, the next one, they are also mostly marine and photosynthetic. So now we are going to just say the characteristics of this. They appear as yellow, green, blue, red depending on the main pigments present in the cell. Now their cell wall is having stiff cellulose plates on the outer surface. Most of them are having 2 flagella. Now if you are seeing this diagram also, you can see they are having flagella. So you can see that they are having flagella. So they are mostly having flagella, and one lies longitudinally and another lies transversely in a furrow between the wall plates. Now there are red dinoflagellates, that is goniologs. So what is this goniologs doing? It is undergoing rapid multiplication, and that makes the sea appear red tides, that is Red Sea. The Red Sea is red in color because of this presence of red dinoflagellates, because of this goniologs, because they are having rapid multiplication and they keep on multiplying so fast that the full sea appears red in color because of this dinoflagellates. Now, at times, these toxins are released from this dinoflagellates, which may kill a large number of marine animals, such as fishes. So, what is this called? This is called PSP. This is a very new term to you all. Please learn it. This is called paralytic shellfish poisoning. So, they are releasing toxins in some cases in huge amounts that can kill marine animals, such as fishes, and that is called paralytic shellfish poisoning. Is it clear? Now comes the third category in this kingdom protista. They are euglenoids. They are freshwater organisms found in stagnant water. Instead of cell wall, they are having a protein-rich layer called the pedicle, which makes their body flexible. Okay, they have two flagella, one short and one long. Now, in presence of sunlight, they are photosynthetic, which when deprived of sunlight, they behave totally as heterotrophs. This is a peculiar characteristic of euglenoids. So, in presence of sunlight, they are photosynthetic. When they are deprived of sunlight, they are totally acting as heterotrophs, and by predating on other animals, they survive. Now, the pigments of euglenoids are identical to those present in higher animals. Now, the example is euglena. Here, I have given you a structure of euglena with one flagella. You can see this is euglena. Okay, this is also a microscopic diagram of euglena. Just go through it, go through whatever slides, whatever notes I have made. Just learn them. You will get a clear idea of everything. So, let us move on to the next one. Now, next comes the slime molds. So, slime molds are, what are they? They are saprophytic protists. Their body moves along the decaying twigs and leaves, engulfing the organic material. Now, under suitable conditions, they are forming aggregation called the plasmodium, and which may grow and spread on several feet. So, this aggregation called plasmodium, which may grow and they may spread over several feet. And during certain unfavorable conditions, the plasmodium differentiates and forms the fruiting bodies bearing like spores at their tips. Now, what these spores possess, they are like true cell walls having, and they are extremely resistant, and they're surviving for many years even under certain adverse conditions. And the spores are dispersed by all air currents. Okay, so these are all slime molds. Now, see this diagram. This is a slime mold. Okay, this diagram is a slime mold diagram. So, this is all, they are all dispersed by, the spores are dispersed by air currents, and the spores are having true walls which are extremely resistant, and they are surviving for years. Now, the spores are being all dispersed by air current, as I have told you earlier. So, this is actually a quite like huge topic which I am explaining. So, please go through it properly, and I'll take the fungi part in the next slide. So, next video only. So, let us move on to the next one. Now, last in this category of protista, we are going to discuss about the protozoans. So, till now we have completed chrysophytes, dinoflagellates, euglenoids, slime molds. So, the last one is the protozoans. So, all protozoans are actually heterotrophs, and they live as predators on parasites and are believed to be primitive relatives of animals. Now, this amoeboid protozoans is the first category of protozoans which are living in fresh water, seawater and moist soil, and they move and prey by putting out the pseudopodia. So, the pseudopodia or the false feet, with that only they move also, and they prey by putting the pseudopodia as an amoeba. And marine form have silica cells on their surface. Now, entamoeba are also parasites. So, it is also having, entamoeba is also amoeboid protozoan, and these are all parasites. Next comes the flagellated protozoans. They are free living and parasitic. They have flagella. They are forming several diseases. Their parasitic form is forming several diseases, and like sleeping sickness and all. So, that example is trypanosoma. Now, next one is the ciliated protozoon. So, this ciliated protozoon, they are aquatic and they are actively moving organisms. Because of the process of thousands and thousands of cilia, they have a cavity or a gullet that opens on the outside wall of the body like paramecium. So, that is ciliated protozoons. Last in this category, after that amoeboid protozoons, after that flagellated protozoons and ciliated protozoon, last one is the sporozoons. These are all diverse organisms that have an infectious spore-like stage, infectious spore-like stage comes in their cycle. So, most infectious is this plasmodium, which is Plasmodium vivax. We all know that is a protozoon, and it is a malarial parasite which causes malaria, a disease which has staggering effect on human population. So that is Plasmodium vivax. We all know that. So that Plasmodium vivax is a malarial parasite which is very dangerous for on human population. So these are all protozoans. So till here we will, in the next slide, we will see just the diagram of these three protozoans and we'll conclude. Now types of protozoan, this one is first one, that is the amoeboid protozoan. We can say this one is the amoeboid one. This one is the ciliated one. And this one is the sporozoon or the sporozoa, and this one is the flagellated. So 1, 2, 3 and 4. Just I am giving you the diagrams, go through it, just see the references and see what the characteristics are matching with these diagrams. So this is all about Kingdom Monera and Protista in detail with ample amount of notes and diagrams. Please go through it and please just go through your textbook, whichever you are following. I think this two part will be very much clear to you all. Till today's episode is completed. In the next episode, I'll be coming with the Kingdom Fungi and the viruses, viroids and lichens, which is to be in your curriculum, and I'll be discussing in the next video. So please do subscribe to my channel and don't forget to like and share my videos. Thank you.